Welcome to your last video of the Thanksgiving break. This video is going to focus on translation. So we've already discussed the DNA to mRNA part of the central dogma. And now we're going to be focusing on the mRNA to protein part, which is called translation and happens in the ribosome. So just to recap where we are right now. Right now we have a mature mRNA that three things have happened to it. It's been spliced, so the introns have been cut out, and a GTP cap has been added to it, and a poly-A tail has been added to it, to help it be transported from the nucleus into the cytoplasm and travel to the ribosome without being degraded by enzymes. In translation, the bases of mRNA will be turned into protein, and the bases of mRNA code for amino acids. And we're about to see sort of how that's done. So essentially this is what we just said. The mature mRNA, which has been spliced, has a poly-A tail and a GTP cap, is made in the nucleus, and then it travels through the nuclear pore, which is right here, and to ribosomes to begin the process of translation. So if you remember, when scientists were just trying to discover whether it was DNA or protein, that was a genetic material, they were faced with a quandary with, like, how can DNA be the genetic material when there are only 4 bases? And proteins has 20 different amino acids. Similarly, same question is, how can only 4 bases code for the 20 different amino acids? And if you take a look, we learned this in 9th grade, is that every 3 bases of an mRNA, so here's a 3-base sequence of mRNA, codes for an amino acid. So for example, the AGU codon codes for the amino acid methionine, or MET for short. The CGU 3-base codon, these are called codons, codes for the amino acid arginine. And so the point is, every single 3 bases in an mRNA molecule codes for an amino acid. Now, this is the code, and these are the mRNA codons. And one thing that's true, and then the AP exam loved this idea, is that these codons are true in every living thing. So for example, in every living thing, the codon CCU will code for the amino acid proline. In every single living thing on Earth, the code is read the same way. Another thing that's important to realize is that there are special codons in this codon table. And one is the start codon, which is AUG. And that tells the ribosome to start reading the mRNA at that point. That's where the mRNA will sort of hop on, the ribosome will hop on the mRNA. There are also 3 stop codons. Okay. When the ribosome reaches a stop codon, it will stop adding amino acids at that point and fall off the mRNA. Another thing to realize about this code is the code is what they call redundant. And what that means is that there are several different codons that code for one amino acid. So for example, if you look at the amino acid proline, one of my favorite ones, is that, I just memorized it, I just know it, is that as long as the first 2 bases are CCC, it does not matter what the third base is, it will code for the amino acid proline. This sort of phenomenon is known as the third base wobble. And what that means in many instances, not all, so if for example, if you see here in this box, the third base does matter. If the third base is A, it's GLN, and if the third base is C or U, it's histidine. So sometimes the third base doesn't matter, but in many cases, all along this column in particular, it doesn't matter what the third base is, it will still code for the same amino acid. Now Ralph, and we're going to talk about this, Ralph says, why is the wobble good? So first of all, the wobble, just to define the wobble, is the idea that in many cases, the third base does not matter. The third base of a codon, no matter what it is, in many cases, not all, will code for the same amino acid. Now why that's a good thing, in some cases, is it allows some mistakes to be made that don't affect the amino acid being produced in the protein, which is a good thing. There's some room for error that may not cause drastic effects in the structure and function of the protein. So that's a good thing to some extent. So the next question is, or what happens anyway, is that the mRNA travels to the ribosome. And the question is, how do the amino acids that the mRNA is coding from know where to go on the mRNA strand? So if we take a look at this molecule, this is the DNA which was transcribed into mRNA, and that happened in the nucleus. And now the mRNA is in the ribosome. And the question again that we're trying to answer is, how do the amino acids know where to go on the mRNA? And the answer to that question is that there's a molecule known as a tRNA molecule. And the job of the tRNA molecule is to find the correct amino acid that corresponds to the codon and bring it to the ribosome in the right place. Now you may ask, well, how does it do that? How does it know? And how the amino acid knows is, how the tRNA knows is the tRNA has on it an anticodon, which is complementary to the codon. So if you see here, you have the AUG, the start codon in the mRNA. Okay? And the start codon was where the ribosomes hop on and start translating. But the tRNA, there's a tRNA that exists that's carrying the amino acid for the codon that has the anticodon to AUG. And what an anticodon means is that it's complementary to the DNA strand. So, for example, the codon is AUG, and the complementary anticodon is UAC. And the tRNAs will continuously match the anticodon to the codon, bringing the correct amino acid to the mRNA in the ribosome. We're going to briefly look at the structure of a transfer RNA molecule or a tRNA molecule. So if you take a look, it's made up of RNA, first of all, and actually the RNA has sort of like collapsed on itself or folded to form this T or clover-shaped structure. And there are two important parts of the tRNA. The first part is the anticodon on the bottom. Remember, the anticodon bonds, or hydrogen bonds, to the tRNA. Not to the tRNA, to the mRNA at the right codon. So the codon that this anticodon would bond to would be UUC. More importantly, is that on top of the tRNA, it will be carrying the correct amino acid that corresponds to the codon, not the anticodon, on the mRNA. That's confusing. Monday in class, we're going to do some practice with this. But the amino acid that the tRNA is carrying corresponds to the codon on the mRNA, not the anticodon. This table over here, if I go back, this table over there is the codon table, not the anticodon table. So the tRNA, just to summarize, has two important spots. The anticodon, which is complementary to the codon, and then the amino acid attachment site, which carries the amino acid that corresponds to the codon that the anticodon is complementary to. The last thing we need to discuss is how the ribosome helps bring together the tRNAs carrying the right amino acids with their anticodon and the mRNA together. In the previous exam, before they changed the exam 4 years ago, you had to know this in much, much greater detail. But we're going to look at it a little bit at least. So first of all, the ribosome itself has a structure. The ribosome is composed of two subunits, a large subunit and a small subunit. And these subunits are made up of ribosomal mRNA and proteins. And again, the job of the ribosome is to facilitate or help the coupling or the bringing together of a tRNA carrying the correct amino acid and the mRNA codon that's on the mRNA strand. There's some question about whether the ribosomes are like really, really prevalent enzymes or actually organelle, because they're made up of both protein and ribosomal RNA. Now, there used to be this whole sort of like to-do about these sites in the ribosome. We're not going to have to memorize what the sites do, but we are going to look at a brief animation on how this works. Just make sure I find my animation. Here we go. Okay, so here we have our tRNA molecule. And we know it's a mRNA molecule. We know it's an mRNA molecule for a couple reasons. First, you see the GTP cap, and you see the poly A tail, and the other dead giveaway is you have U's, so obviously you know it's an RNA molecule. So we're going to click play, and you'll see the first thing that happens is the small ribosomal unit, or the small segment of the ribosome, hops onto the mRNA. And then a tRNA comes in. Notice that at the start codon, where the small subunit has bound. And if you notice, the anticodon is complementary to the codon on the mRNA. And it's carrying the correct amino acid met. Now what's going to happen is the large ribosomal subunit is going to attach to the small one. And then the next tRNA that's complementary to the next codon, or has the anticodon that's complementary to the next codon, is going to come in. So you see here we have the codon AAG, and then the tRNA with the anticodon UUC comes in this area in hydrogen bonds, and it's carrying lyse, which is the right amino acid for the codon AAU. And what you're going to see is going to happen is that this amino acid is going to be transferred to this one, and they're going to peptide bond. Then this tRNA is going to leave, and the ribosomal subunit is going to slide. Or it's going to slide and then it's going to leave. The order is not necessarily important. And what's going to happen is the next tRNA is going to come in. That's complementary to the codon UUU. Here's the anticodon AAA, and it's carrying the amino acid Phi, which corresponds to the codon UUU. It's going to transfer, form the peptide bond. The ribosomal unit is going to slide, and the tRNA is going to leave. Next, tRNA comes in with the codon-anticodon pairing. There's a peptide bond formation. tRNA leaves. The next tRNA comes in. Now here, if you'll notice, we don't have another tRNA coming in. We have a release factor binding. And the reason why we have the release factor binding is that we've reached a stop codon. UAA is a stop codon. And once the release factor binds to the ribosomal subunit, that causes the release of everything. And at this point, the proton is a release proton. This is a really small protein. Protein will fold, and the ribosome can go back to the beginning and start translating this protein again. Now, that is essentially the end of the lecture. I would make sure you answer the guided questions that are associated now. On Monday in class, we're going to do some activities to practice this entire process, because we've gone through a lot at this point. But answer the guided questions, and I will see you guys all on Monday.